having weren't exciting enough. We're really getting to the part, and I'm being serious, that is the most exciting. At least I remember when I was a student, this sort of brought together things that we have discussed in so many different ways. We've talked about the structure and function of the gene from a lot of different perspectives, and now we're really at that point where the molecular underpinnings that really make it happen are going to be able to be understood. If you're following along in order, we've been talking about in this section the central dogma and the fact that DNA is storing information in its base pair sequence, and that a messenger RNA is being copied through transcription. Um, Ultimately, that information is going to be brought out to be made into a protein. So the idea is not revolutionary. Beadle and Tatum said one gene, one enzyme, or one gene, one protein a while back. Now we're going to see how it literally and actually occurs. How does genotype, meaning the actual, in this context, meaning the actual sequence of bases in a DNA molecule, end up translating into phenotype, which means the actual amino acid sequence that creates a particular type of protein that does something. So before we get into the mechanics of it, we need to talk about the genetic code. So let's do that. Now, you see here I wrote translation and the genetic code, the construction of a protein. This is our goal, if you will. We're going to take information that was in messenger RNA and use it to make a protein. And this process is called translation. The reason it's called translation, perhaps obviously, is that we're changing languages here. Remember, copying DNA into RNA is transcription. That has the idea of just copying it. A scribe just copies. Now, it's not identical, but both DNA and RNA are speaking nucleic acid language, right? But now we need to translate that information, that language, into the language of proteins, which is using amino acids as its basic words or letters, depending how you look at it. So translation most normally, and I think we've said this before, is used to refer to the step whereby the information in messenger RNA is made into a protein. Some people, when they say protein synthesis, they mean this whole thing, but to me, protein synthesis is broader, and it means all of the steps, including transcription and translation, getting you from the DNA information to the protein information. So let's recap a tiny bit here. When a mature messenger RNA has been released to the cytoplasm, so the implication here is that this would be happening in a eukaryote only because that was the last thing we were talking about. But whether bacteria or eukaryote, you have to have, I mean, there isn't a lot of maturation and processing in prokaryotes as we've discussed, but one way or the other, we're going to have a messenger RNA that's, quote, ready to go in either case. And the same goes for tRNAs and ribosomal RNAs. We need all of these in order to actually get the protein synthesis going. We'll talk about the mechanics in the next part, but remember that ribosomal subunits actually start getting put together in the nucleus, and that all that transcription of ribosomal RNAs is actually what the nucleolus actually is. Ribosomes, remember, have two subunits, a large one and a small one. In bacteria, they're 30S and 50S. Remember that S for sedimentation coefficient. We talked in an earlier lecture, or I should say theory session, about 16S ribosomal RNAs, etc. So when they're all put together, you get subunits with this. They're put together with proteins as well, ribosomal proteins. You get subunits of these sizes. One small, one large. Okay? But I don't know if the word philosophical is the right word. But the whole point of this theory session is to think, and I can't be a positive, but I think scientists of the day, before they really had the hard experimental facts, they were thinking about it. Remember, Watson and Crick really didn't do a lot of experiments. They thought about using data that people had, what would make sense in terms of DNA structure, just as one example. So before we start doing smart experiments, we have to think about the genetic code in this case, because from everything we know, and when I say we, I mean both we in this class, but also scientists up to this point, probably in the late 1960s. The idea of a genetic code, I say, is implicit because we've sort of said this, but what I mean is somehow we know information has to be converted from nucleotide language, which DNA and RNA speak, to amino acid language, and that's the idea of translation, as we've said. So let's just think about this. In any code, guys, a language is a code. A code can be so many different forms, but by the definition of a code, one or more symbols, whether that symbol's literally one symbol or a word or a picture or whatever it is, one or more symbols in one language have to represent a corresponding symbol in the other language somehow. If you're translating English to French, it's not a great analogy, but we know, maybe you speak French, but a chair, I point at it, you go, yeah, chair. In France, they say chaise, I think. In other words, we're referring to the same thing, but there's one word for it in one language and one in the other. Not the best example, but you get the idea. So somehow, there's the bottom line. There are 4 nucleotides in the nucleotide language, because remember, what they really are A, G, C, and U, because it's the RNA that's going to be directly translated. But 20 amino acids, there are 20, and there are necessarily 20. They have to be specified. So you can clearly see, we're going to talk about this for a minute, it can't just be a one-to-one correspondence, but no one said it was. I mean, at this point, this is what's fascinating about it. However this code came into being, which is a much more philosophical discussion, imagine the excitement of trying to figure this out and having the tools to do it. So here's a table that I think, if you feel like I'm just babbling and you're getting lost, I hope you don't feel that way, but here's something to ground us, okay? Because look, here's a table that shows, if we're just doing a thought experiment now, we're saying, look, if it were true that the number of bases in the genetic code that specified one amino acid was, in this case, 1, I hope it makes sense to you that the maximum, the exact number of amino acids that you could specify would be 4, right? Because there's 4 different nucleotides. So this would mean, it's not correct, but it would mean, say A means methionine, G means aspartic acid, whatever it is. You'd be able to specify 4 if there was a direct 1-to-1 ratio here. That's not happening. You don't have to do any experiment to realize, that that's not feasible. It's not reasonable. So they started to think, well, what if it's a doublet code? A doublet code, which I'll write here, would mean that a pair of nucleotides would act as a unit in specifying one amino acid. So for example, AA would be one doublet, which would mean something. AU might be another doublet that would mean something. It probably doesn't take a lot of math skills, no offense if you're not great at math, to realize that if you did it that way, there would be 16 different combinations of 4 nucleotides arranged into pairs, each of which act as a unit. There's a formula for this, actually, if you want to remember it. It's 2 to the n, and what that means is where n equals number of bases acting as a unit. So if you just want to make sure we did our math right, when one base is acting as a unit, 2 to the 1 equals, I'm sorry, I messed it up, didn't I? Rather than start all over, we'll just say apologize, and it's not 2 to the n, it's 4 to the n. I'm sorry about that. So if n equals the number of bases, 4 to the n where n is 1, I really apologize for that confusion. But 4 to the 1 is 4, right? So that's where we get this from. 4 to the second power, which is 4 times 4, is 16. I don't know why I screwed that up, but the bottom line is the 4 comes from the fact that there are 4 different bases, okay? When you raise it to the n power, that tells you how many combinations you're going to get if they act as a unit. Sorry about that. But now that we understand it, when you have 3 here, which would mean the code was triplet, before we do the math, let's see what that would mean. It would mean AAA was one unit, AAU was another unit, AAG. You don't need the formula perhaps to envision it, but if we use the formula and say 4 to the third power, we find out that if we do that math, we would have 64 possible number of different amino acids that can be separated. Now look, let's take all this and let it roll around our head a little bit. 16 is close, but it's not good enough. We need 20, it's a fact. So the doublet code would almost give you enough combinations to get 20, but it's close but no cigar, as they say. So the triplet code would give you 64. Now that's enough, but there seemed to be a lot of extras, so that was a bit of a question. But logically speaking, it seemed that a triplet code was the minimum that you would need in order to be able to specify all of the amino acids. It's not proof. You could say, well, what about a quadruplet code where 4 bases act? That would give you, I'm almost afraid to do the math, 4 to the 4. That would give you 256 different possibilities. You only need 20. Now, there's no law in the universe that said it's impossible that that happens. But maybe you guys have heard of Occam's razor, which is basically an old principle that says unless you have evidence to the contrary, don't overcomplicate things just for the sake of it. There's a few ways of phrasing it, but basically it means, you know, we're going to do experiments, but just to look at the logic of this, a triplet code seems reasonable. We're going to have to figure out what happens with those other 44 combinations. Okay? Now, some of this we may already have said, but logic dictates that the code must at least be a triplet codon, a triplet code in which a sequence of 3 nucleotides acting as a unit, and this is an important term, this is why I just said it, a sequence of 3 nucleotides in the messenger RNA that acts as a unit to specify one amino acid is referred to as a codon. That's very important that you make clear that the codon is not in the DNA, it's in the RNA, and it's a sequence of 3 nucleotides in a messenger RNA molecule. Now, armed with this idea, experiments that were really clever, and I'm not going to say they weren't hard, but the idea behind it, it just makes a lot of sense. Now, I'm emphasizing Crick here, and I love Crick. He was certainly part of all this. There were certainly other individuals prominently playing a role in figuring all of this out. So they get to be Crick's colleagues. In the 60s, they used frame shift mutations, which I think we've mentioned before. But the bottom line is this. This is what they found out. They found out that if you add 1 or 2 nucleotides, or delete them for that matter, from a sequence, a mutant phenotype occurs. But in many cases, if 3 or multiples of 3 are added or deleted, the wild-type phenotype usually persists. That is extremely strong evidence that the code is, in fact, a triplet code. We can draw a quick thing to see why. Say, I'll make a new one. We're sick of A's. CCC, we now know, means the amino acid proline. But let's say in a normal sequence, the next codon was AAA, right? So I think this will work. If we delete, so the whole sequence here would be CCC, AAA, going on in all directions, but we're focusing in on that part. If we delete this, right, it messes everything up. And the reason really is that we've shifted the reading frame. It's now being read as CCA, okay? The same logic goes if we get rid of 2, and the same logic goes if you add 1 or 2. I don't need to draw it. But now we're reading this as CAA, right? Which is a mess. But so what happens if we get rid of all 3? Well, that codon may be gone, and the amino acid that it codes for may be gone, but it doesn't throw off the reading frame. Everything else stays okay. So that's the short version of why these type of experiments, proof is a very strong word, but they give strong support to the idea of a triplet code, which is going to be given a lot more support in just a second. So if we get the idea in our head that it is a triplet code, as we just said before, it seems now that there's too many codons. So we have to ask ourselves what's going on with that. So a lot of experiments, and as you can imagine, a lot of people got excited about not only saying it's a triplet code, but figuring out what all these triplets mean. Actually, it's like breaking a cipher, figuring out something. It's not something human made, it's something that exists in nature. So it turns out that the genetic code is degenerate. That word has some implications in the English language, but what it means in this context is that it turns out that more than one codon can specify the same amino acid, which means there's some redundancy if you want to think of it in a different way. We'll see what that means in a minute. Now, the code was actually cracked in the late 60s using what's called an in vitro protein synthesizing system, along with some synthetic messenger RNAs. So that's a mouthful. It's not by any means easy to physically do, but the idea of it isn't so bad. The idea is you can make a messenger RNA, and today it's easy to do so. In those days it was a little harder. But you can make one, for example, that just says, okay, here's a messenger RNA, A, whatever sequence you want in theory, but one with all the same nucleotide was a little easier to do. That's a messenger RNA that could be translated. And they made these cell-free translation systems that contained ribosomes. They contained all the amino acids, and they basically threw a particular mRNA in there and said, let's see what happens. Let's see what kind of a protein or a polypeptide comes out. So I hope you follow this logic. If you put a poly-U nucleotide into such a system, and that just simply means it's literally U, the actual size doesn't matter. Longer is fine. That's all that's in there. And then if you isolate proteins, not amino acids, you isolate the protein that's made, it turns out that one protein gets made, and it's polyphenylalanine. It's really, simple. It is simple. Doesn't mean it's easy. I wouldn't have wanted to have to do this, although it would have been fun. That means that UUU must mean phenylalanine in the code, right? Because think of the logic. Whatever the reading frame is, if you make one like this, any possible way it was read, the only codon that could possibly be viable is UUU. It's all that's there, right? So they started to do some detective work, and they said, all right, UUU, phenylalanine, write that down. Then we'll do one that's AAAAAAA, and we'll see what that means. By doing it that way, you can figure out 4, right? We don't have to go through the entire logic of it, but if you make an alternating one like this, if you think about that, there's only 2 possible codons that there could be, right? AUA and UAU, because any way you look at it, those are the only 2 triplets that you could come up with. So they then find that 2 types of polypeptides are made there, or I should say a polypeptide that's alternating in amino acids. And by process of elimination, you can actually just start figuring out all of them. It's really exciting, in case you didn't know. It turns out, guys, I cannot overemphasize this. The genetic code, I say practically, there are some minor exceptions, but overwhelmingly the genetic code is universal. And what that means is the same in all organisms. Just think about this for a minute. This was actually done using E. coli bacteria, just because they're so easy to work with. And we've mentioned that on occasion, that often microorganisms are used. So when they found out and they figured out the whole genetic code in E. coli, it was not 100% clear that a giraffe or a plant, or a human being for that matter, would employ the exact same code. It's obviously not the same, but there's a couple of hundred languages that people use on the earth. Who was to say that only one universal language exists in biology? It's a big question, both philosophically and factually. It turns out that there is one language of life. And, this may not be part of the course, but it's things that I know I was thinking about at the time. So let me just say that, people interpret this one of 2 ways. Clearly, all living things, from a bacterium to me and you, we have something in common. And I don't just mean the obvious, that we're alive. We have something in common in the deep instructions and the process by which we are made. Now, a lot of people look at that as evidence of an evolution from a sort of single individual system that then spread out, but the language never really changed. Um, I almost hesitate to say this, but it's a great analogy. And some of you guys out there like Star Trek. Don't pretend you don't. If you don't worry about it. But when Star Trek was, it's still popular, I guess, but in the early days, before my time even, it was a reasonable question to ask. How come we go to all these planets all over the place and pretty much people all look the same? There were a few exceptions. They said they had universal translators, that they were speaking different languages. But it seemed awful odd that pretty much everyone looked like a human being, whether they had a little different skin tone or facial hair or something like that. And later on in the series, they addressed that. And there is a point to this. I don't remember the exact specifics, but the idea was that some master alien race had sort of taken the general idea of a human being and planted these seeds all across our galaxy. So it wasn't just coincidence that everybody looked relatively much the same. It was because they all came from a common ancestor. And that's the idea of evolution, that all life forms came from a common ancestor. And that's one explanation of, why the genetic code is the same in all organisms. Or I should say, it's an interpretation that explains why that would be true. Now, of course, there are some people who say, and it's not for me to judge, that if there is a designer of this code, then that's the language they chose, and that's why everybody has it. So you're going to have to figure that part out as you progress through your thoughts and your studies. But the fact that it's universal has a lot of practical significance. And what I mean by that is, imagine, now that it's not true, nobody worries about this, but imagine if it was different in every organism around, or just mammals had one way, birds had another. We'd have to do this over and over and over again. We now know it works the same. There are massive implications in this for genetic engineering. You take a human gene, pop it into a bacteria, they all do it the same way. Not identically, but generally speaking. So universal genetic code is really big. Now, memorize this. I'm half kidding, but if your professor, I don't want to say mean, but let's say if your professor is particularly something, they want you to memorize this, I guess you have no choice. But in most cases, you don't have to memorize it. Why I'm showing it to you is a couple of reasons. This is, in fact, the end result of all that experimentation about what do each of the triplets mean. You'll see if you go across here that there are 64 boxes here that correspond to the 64 possible combinations of 3. You could take a look at the table. We have the first base here, the second base up here, and the third base over there. So at any place where these 3 intersect, you're going to get a unique codon, which it says right in the middle, so you don't have to think of it that way. So let me show you a couple of features of this. It's good to memorize a couple things. The start codon always is AUG, you guys, all right? And that happens to mean the amino acid methionine as well. So if you were going to memorize any of these, I would say the start codon would be a good one. Then there are 3 stop codons over here, UAA, UAG, and UGA. Those are 3 stop codons. I can't tell you why there's 3, but each of those says stops. We're going to get into the process next time. But remember in transcription we had the promoter saying start here, then elongation, then we had a terminator saying stop. It's sort of the same idea here. The start codon perhaps obviously says start here when you're making your protein. And the stop says, okay, you're done. Stop there. Now, what that means is, guys, the stop codons do not code for any particular amino acid. They just code for nothing, which is basically why they say stop. That means that there are 61 of these things that are being utilized to code for amino acids. Now, sometimes it's unique, like in the case of methionine there. But let's just take, we're not going to look at everything, but let's just look around. Phenylalanine, right? I told you poly-U meant phenylalanine, but let's check out what else might mean phenylalanine. Well, there are 2 codons there, UUU and UUC, both of which mean phenylalanine, right? So this is an example of the degeneracy of the code. If you look around, you'll see that those are the only ones that mean phenylalanine. But in a particular example, so let's look here. Leucine, there are 4 codons there that code for leucine: CUU, CUC, CUA, and CUG. But if you look around, you'll find that those are actually the only ones that code for leucine. So there's no absolute way to know like how much redundancy is there for a particular amino acid. I'll show you another one here. I just want to look around. All right, so serine has a situation similar to leucine in that there are 4 there. But then over here, seemingly just out of the blue, there's AGU and AGC that also means serine, right? So you have to, this is what you have to do experimentally. There's no way to think about this and say, you know, CCC, I bet that means proline. There's no logical basis for that. It's almost like saying, well, that'll probably ruin it, so I won't say it. But, all right, so, you know, as I said, you don't have to memorize this as far as I'm concerned, but I'm not the one you have to please, although I hope you'd like to please me. Your professor obviously is the one you have to please. So if they say memorize it, I don't know what to tell you. I really don't. It's unlikely. In most cases, you're provided with a copy of this table during an exam, or certainly if you're doing homework problems, you'll be able to refer to this or something like it. Before we move on, take a look at something that may already have caught your eye. In these cases, like over here, where there are 4 codons that all mean the same amino acid, it probably didn't escape your view, but if it did, let's look. What's the same about all these? The first 2 letters are always the same. And what that means, guys, is that CU anything, in fact, means leucine, right? CUU, CUC, CUG, CUA. What that really means is that the third position, is irrelevant in that particular case. And if you really peruse the table, you'll find that that's not an isolated situation. So this is important, and it led, again, Crick is very prominent in these things, the proposition of what he called the wobble theory. So he said that it explains some, not all, of the degenerate codon's ability to code for the same amino acid. Because, as we just looked, it seems that the first 2 letters of a codon, in many cases at least, seem to be more important than the third one in determining the correct amino acid. We didn't get into this deeply, but there are certain modified bases, inosine being a popular one. If they're present in the anticodon loop, one tRNA may be able to read 3 codons for an amino acid. So the bottom line is, it's not always true, but the first 2 letters are oftentimes enough to specify the correct amino acid. Remember, I hope this will go together well. If you look back at that table we said, if it was a doublet code, which is almost what we're saying here, you'd have 16 amino acids that you could specify. So what that means is, in a certain sense, guys, I hope you understand this, part of it is almost like a doublet code. The third one's just getting thrown in there because we need to have a triplet code to have enough. But as long as we're going to have extras, they can come in handy because, you know, the opposite idea would be to just use 20 and the other ones don't mean anything. It means, though, a couple of things. First of all, it's considered to be a fail-safe in case of mutation. It wouldn't always work, but the point being, if a codon like CCC mutated to CCG, you would think that would be a problem. But if we go back and look, CCC is proline and CCG is also proline. So I know that there are other types of mutations that can screw things up, but something's better than nothing in this case. So what I mean is a mutation in the third position of these type of degenerate codons is not going to have any effect. A mutation at the DNA level will not have any effect at the protein level for those particular ones. It also means, guys, that there do not have to be 61 tRNAs present for translation to occur, because you don't necessarily need one for every single codon. In a situation where like we just said, CC whatever is enough to mean a particular amino acid, a tRNA we don't need a corresponding tRNA for all four of them. We'll leave it at that. I realize that we're sort of bridging into some information that we didn't fully discuss yet. There are anywhere between 40 to 60 tRNAs in most cells. It depends on the species. It depends on a lot of other factors. But think about this, though. Absolutely, there would have to be at a bare minimum 20 tRNAs, right? Just not experimentally, just logically. You'd have to have one for every amino acid at least. Because this degeneracy is not consistent and it's not the same with every single amino acid, there's going to have to be more than that. But you don't need one necessarily for every single codon. All right? Now, as a sort of bridge from this session into the next one, where we're really going to go into the mechanism of how this works, I think I've showed you either this picture or a picture like it before, but we're going to put these pieces together into what we have been talking about. And really, if you can understand this, you're going to not have any problem understanding how translation literally occurs. I once referred to a tRNA molecule as a decoder molecule. And the reason is, okay, so let's just orient ourselves to what we're looking at here. Down the bottom here, this is a messenger RNA, and each of these is a nucleotide, right? This is a base right here. Now you can see some of them have two, which means they're purines and they're different colors, and some have one. So all four bases are being represented there. Only three of them are labeled UUU. You may remember UUU means phenylalanine, right? So you can experimentally show that, but what does it mean at the molecular level that UUU means phenylalanine? This is what it means. There's a lot of means in there. There's a transfer RNA, and they're called transfer because they transfer amino acids. There's two functional important parts of tRNAs. There's more than two, but there are two main ones. And I think we've discussed them before. But this is the amino acid acceptor stem. It's always got the sequence CCA. And remember, this whole thing is a stem here. It's not a stem loop. So that means an amino acid gets attached up there. Then we have the anticodon loop down here. It's sort of made in yellow, but remember, a tRNA is an RNA molecule with one base after the next, one nucleotide after the next. It's just folded up into an interesting clover leaf-like secondary structure. But there are three RNA nucleotides down at the bottom here, which comprise the so-called anticodon loop. This, if you didn't think of it, that's a codon, right? Three bases in a messenger RNA that are going to code or specify one amino acid. So the whole key to it is why tRNA is a decoder molecule. At the anticodon loop, it's, quote, speaking nucleotide language. It is an RNA. And base pairing rules are relevant here. So if a codon in the messenger RNA says UUU, automatically, because of the base pairing rules, a tRNA with the opposite anticodon, AAA, is going to be attracted there. And we're going to explain why later. But just remember now, this is how the connection is made. Every tRNA with an anticodon of AAA, it always carries the amino acid phenylalanine. That's how we can say that UUU means phenylalanine. It's mediated through a specific transfer RNA with a definite anticodon loop that corresponds to the codon, and then it's carrying this amino acid on its other end. It's a dynamic process, but if you understand that idea, you're going to be able to understand the molecular basis of translation. Thanks for spending this time with me. I can't wait to go ahead and start talking about the actual mechanism that puts all of these somewhat abstract ideas into practice in a living cell. I'll see you then.